ability to study phase polyphenism, which is the phenomenon responsible for the swarm formation in locusts, and the ability to prevent plagues of locusts and the agricultural destruction that they cause in various areas around the world. Relies on a deeper understanding of the phenomenon. It's not extremely well understood, and to be able to understand the transition from non-swarming to swarming, which causes the agricultural destruction, will improve our ability to prevent it. So now that I've given you an overview of the background of this project, I need to start with. No, I don't. So I proceed to explain locust phase polyphenism, which is a type of something called phenotypic plasticity. Phenotypic plasticity is a physical change in an individual due to changes in their environmental conditions. So two individuals of the same genetic background may appear or act different due to a difference in environment. And for locusts specifically, this is density dependent, meaning it relies on the population density that they are living in. So locusts who are solitary with cryptic coloration, green and brown to blend into their backgrounds, will transform into gregarious, brightly disruptive colored individuals when their population density is high. And then this leads to swarm formations and, again, agricultural destruction. So the species that was used in this study was specifically the desert locust. This locust is located in the Middle East and Africa, but it's relevant to all locusts. It's known for the biblical plagues to show the level of destruction that they can cause, and it is the best studied species of the genus that locusts are present in. It occupies the basal position of the phylogeny, meaning that most of the locusts of this genus are evolutionarily related to it, so the traits of swarming are relatively consistent between all of them. And phase polyphenism is present in all of these locusts, like I said. So to continue on, why are we using Raman? So traditional techniques used for analysis of phase polyphenism are chemical analysis and behavioral assays. Both of these tools have their downfalls, and Raman may be able to improve these experiments. So chemical analysis is usually just to determine the molecular underpinnings of phase changes. So we look at chemical differences between the two phases to see what is present or what changes when they form swarms, become gregarious. These studies have also explored genetic expression and evolutionary connections between locusts of the different species, but these experiments often are expensive and very laborious. The other type is behavioral assays, where an individual is usually placed in an arena to test for what phase it is in. These normally follow the original procedure, in which there's a rectangular arena with two chambers on opposite sides, one having various crowded individuals and the other having an empty chamber, and whichever locust or whichever side the locust tends to is often the determination for what phase they're in. These are lengthy, as you need to watch the entire process or record it, and using Raman would prevent the long time consumption of this and the expensive labor of chemical analysis. So to give a bit of materials and methods for what this procedure was, I mainly performed analysis throughout the summer, but there was also collection of the grasshopper samples. So the grasshoppers were raised in both isolated and crowded conditions to determine their phases. The grasshoppers were then collected and marked for the conditions that they were raised in, including the sex, the isolated or crowded conditions, the diet, which was a wheat diet, their age. They were taken at 1st and 6th instar, and their species, which for this was just the desert locust. And 20 microliters of hemolymph was collected from them, which is important because this allows us to also release the grasshopper back into its environment, potentially observing it later. The analysis was done using a confocal Raman microscope. This has a 480 nanometer laser with a power of 1 milliwatt and an acquisition time of 1 second. 25 spectra were gathered per sample, resulting in 2,500 from the 100 total samples used. And they were uploaded into PLS Toolbox and MATLAB for analysis, and the raw spectra were processed to then be performed through analysis with partial least squares discriminant analysis, PLSDA, to discriminate between the two conditions, and then identify key peaks at the wave numbers shown here, were then analyzed using Kruskal-Wallis one-way ANOVA, which is a test to determine the significance of the peaks between two groups. So I have a couple of pictures here just from what was going on. I have the tubes that had the samples in them, how they were labeled. I have a picture of the laser from the confocal microscope that we use. The laser then goes in the microscope and is recorded through a camera and outputs the spectrum, which you can see on the program used on the computer. So for my results and discussion, I first want to start with the spectra of all of the spectra we gathered. The spectra were averaged by group for crowded and isolated. They were baseline corrected and then normalized, and it gives the spectra that I have here. There are a couple of peaks marked, but the notable peaks that were used for analysis are at 1008, 1162, and 1530, which all are identified as carotenoids, an organic dye that is often contributed from the diet that they eat, as well as 1196, which is a phenolic compound, again, present in the diet they eat. This phenolic compound is present in cell walls of plants. So next, I wanted to talk about PLSDA. So what PLSDA does is we input all of the spectra, and it discriminates the individual spectrums based off of the differences in each spectrum. The total are looked at, and then for each individual one, it assigns it to the classes that you choose. In this case, crowded and isolated, there were only 2 used. And then it reports information on how accurately it is able to predict those spectra as what they are. So the information that's used for this study that is reported, there's a lot here, but mainly what we look at is the TPR, which is the true positive rate. This is the amount of spectra that are accurately classified as the class that they are. So the ones that are crowded or determined as crowded that are crowded contribute to this value, which was 88% or 88.2% for crowded and 89.4% for isolated. The other information that's also relevant is the F1 value, which reports the ability of the model to accurately predict a class while also minimizing the false positives, which FPR is the false positive rate. And then the last value that's important is the Matthews correlation coefficient. It is the reliability of the model overall. It can range from -1 to 1, -1 being all spectra determined as negative, or like determined incorrectly, and 1 being all spectra determined correctly. The other values can be relevant, but since we're using 2 classes, most of them are repetitive of the information before. So the last test that was performed was the Kruskal-Wallis one-way ANOVA. The Kruskal-Wallis test is a non-parametric test, meaning that there is no normal distribution required to perform this test. And what it does is it ranks the mean intensities of peaks. And then for the classes that were chosen at specific wave numbers, it shows the ranks of those 2 peaks to determine if there's a statistical significance and a difference between them. If you look at these, you can notice that for all of these, there's a very big difference. And for all of them, also isolated is higher than crowded. I'd like to mention that because all of these compounds are located or are likely from the diet, this could be due to dietary conditions of crowded species, potentially competition for resources, causing them to have lower intensities of the chemicals or less going into their diet and then into the hemolymph, which I think I forgot to mention earlier is the equivalent of the insect blood. Also that because all of these are significant, we could use all of these peaks potentially in the future to identify them as isolated or crowded. So now that I've explained all of my results, I want to go to final conclusions, which is that the Raman spectroscopy has proven to be successful from this data. It achieved high accuracy rates of 88.2% for crowded and 89.4% for isolated conditions. This is overall an 88.8% accuracy, which is very good. There was significance observed at all 4 of the peaks that I looked at, meaning that they can all be used for identification. And also, like I mentioned, this could be due to dietary conditions, which is likely what's causing them to go into swarming, which is known before. And then there's potential for further applications. So it can be extended to analyze different conditions depending on rearing density, such as looking at different types of species. There is a sister species of Central America, which is closer to here, that is very applicable from this data of the desert locust. And we could look at that species and see how these conditions relate to them, and also specific subgroup conditions. So looking at information within the grasshopper and how it's connected to the phase transitions can be analyzed further now that we know that Raman is applicable. And that's going to be the end of my presentation. So I want to give a thank you to everyone. I didn't mention them on the slide, but the collaborators, Delia and Gregory Sword, who reared the grasshoppers for the study. All of the undergraduates who participated in this program with me, some of them helping me. Aiden helped me a lot with this project. I learned a lot from him. also wanted to thank Dimitri as the mentor and advisor from you throughout this program, as well as all of the CBTS and DHS officials who made this possible this summer. It's been a really great and fun experience, especially Heather and Chris, who I've seen multiple times when we've gone to different presentations and throughout the program, as well as Dr. Mary Breik, who helped us with the papers and presentations and all of the guidelines for putting this stuff together in the end. So that is my presentation. Does anyone have any questions?
So examination of grasshoppers required kind of some dissection then?
So
Always?
the hemolymph was gathered with a needle, so it was just put into the bag. We were able to take the hemolymph. There was no like dissection. Like I mentioned, it's very non-invasive. We can really hook it back to the wild with minimal interference. So just some hemolymph is taken, put into a capillary tube, and then studied later.
Can I ask
Yeah, you can ask questions.
Maybe you can elaborate more between on the connection of your research and national security.
Oh, yeah. So like I mentioned, these locust species have the ability to form swarms and plagues. They can cause like massive agricultural damage. It's been observed in the past, and there are a lot of regulations to put to try and stop locusts from spreading and swarming in the wild right now. These species can cause agricultural damage. They are also very near to America, like the Central American locust that I didn't look at here, but is still applicable as it's very related to the desert locust, is just under the border of the US. If these species were able to get in and be like unsupervised and cause swarms, this could cause agricultural damage, causing massive issues.
Do locusts carry any diseases or pests or any other diseases, plant diseases?
I'm not sure about that one. I just know about the swarm formation because that's the main thing that's being focused
Yeah,
on.
for sure destructive by themselves.
Yes, they're very destructive on
They can eat the
their
whole, the entire fields.
entire fields. And the transition from isolated to crowded can be very quick.
Yeah.
Once they are all in a crowded area, they start to change into swarms. It was actually thought that before the swarming locusts and their isolated forms were different species, but it's recently, or within the past like century, they were identified as the same species, and that's how these studies emerged. That's all for questions. So thank you again.
Okay.
What's next? You want to alternate Westlaco and? Volunteer from this today. Fine, fine.
You're next.
Next. Okay, so again, I'm going to share again.
Oh, is it the same as what you said? Oh, okay.
Didn't make a change.
No worries.
So.
There's one this side. Not a real scientist unless you didn't change it.
So now sharing it.
Yeah.
I saw it on.
Yeah.
Yeah, I can do that.
Huh? She's not sure. I'm not school that other group.
Oh, then here. Yeah.
She has to tell us that she was our like
Yeah.
Which one's Max?
it's one at the very top.
Reading? You guys can see the slides.
I think they just did a speed drive.
I'm gonna try, I'm sharing and sharing.
Oh, and then maybe not.
Oh, yeah, share the PowerPoint. Oh, with it, yeah.
Yeah.
She got it. Oh, you got the TV working.
The TV, yeah. This one
What's up?
Get close.
Wow. Keep on speaking.
You're good.
Yay! All right, slideshow. All right.
Hello, everybody. My name is Maximiliano Hernandez. I am a biomedical science major at UTRGV in the Rio Grande Valley. And I'm going to be talking about citrus greening disease and how we can identify and develop resistances towards sea lice infection. This is a little outline of what we're going to be talking about. Basically, citrus greening identification, rhizobia mediated transformation, hairy root identification, and hairy root diagnostics, which we're going to go over a little bit later on. A little bit of background information on what the citrus industry is here in the US. So Florida, Texas, Arizona, and California, they lead the way for all citrus plants, with Florida being the biggest producer of citrus. So 42% of the citrus as of 2019 to 2020, we have the third largest exporter of citrus plants, of citrus fruits, with Brazil and China being the leading countries that beat us in terms of our exportation. And our US citrus cultivation, so 7.78 million tons of citrus is usually on average produced. This is a range as of 2019 to 2020. However, you can estimate how much we actually produce, and we can see that just with the US alone, it is quite a bit. Now you can just imagine what Brazil and China must be producing with all the land that they have. So here we have a chart. On the left-hand side, I had put a specific type of citrus that we produce here in Florida. So the sweet orange production, you can see that a couple of different countries that produce sweet orange are labeled on the left. I implemented this graph because I wanted to just give like a visual of like the numbers of how much production is actually going into the citrus industry. So this is just with sweet orange alone. And then you can just imagine with, let's say, grapefruit, rough lemons, which is also something that we work with at the AgriLife Extension. On the right-hand side, we have citrus production in the US. This is just a graph to visualize our 7.7 million tons. So on average, we can see that it ranges from 2010 to 2020, and it's kind of lowering on this graph, which we are going to explain a little bit on the reason why we presumably see that reduction in values over the years. So on the bottom, we have our citrus value production, and this is estimated at around $3.5 billion in the US. Over the years, it really doesn't fluctuate too much, but that's the range of revenue that is produced as a result of our citrus industry. A little bit more background. Now we're going to start talking about the actual disease and the actual vectors of this disease that cause citrus greening in our citrus-related plants. So here we have the agent citrus psyllid. We'll call it ACP for short. On our right-hand side in the top corner, you can see how these little white squiggly lines on the branch, those are the little nymphs. Those are basically the larvae that grow into the big insect on the left-hand side. So this is what an agent citrus psyllid would basically look like. There's also potato citrus, African potato psyllids, but mostly I'll be focusing on the Asian citrus psyllid side. They both basically do the same thing, but just on different plants. Our species that is the reason for why we have citrus greening is called Candidactis liberbacter asiaticus, CLAS for short. This is the pathogen, the microbe that infects the citrus plant and causes a whole variety of different complications as a result. The vector, the official name for the vector, the scientific name is Diaphorina citri. And as of 2004, we've seen our initial presence of these psyllids and the Candidactis liberbacter asiaticus in Florida. I believe this is the original official timeline for when we had CLAS exposed to the US. And here's a little bit more information. A little graph that shows how Florida is basically the leading producer of citrus and how the CLAS has infected the citrus plants over the years. So we do have an average yield loss on the right-hand side. This has been updated since 2023, and we can see that as of 2023, we have a 45% yield loss. Around 80 to 90 infected trees are present, and especially with Florida in particular, they've had the biggest resurgence of CLAS in the entire country, and they've actually been the ones that got hit the hardest with this pathogen. A little bit more graphs that I've implemented here. This is just another visual, how we see that over the years, the decline in orange production. Specifically, orange is the one that I chose because it had the most visual decline. So over the years, as of 2010, we can see that thousands of acres, about 400,000 acres, 400 acres of citrus plants, specifically oranges, were on a decline as of 2010 to 2020. Here are the tons that were produced in the United States of orange production, which have also shown a really big decline. So our objectives for researching CLAS and for preventing citrus greening disease, here with the research department that I worked with, we used identification methods, specifically molecular identification methods as well as visual. We use diagnostic methods to screen for resistances and enhance prevention against the treatment of citrus greening. And we also, other objectives that we have is to understand the functions of current tools to better improve different aspects of why the pathogen is so difficult to work with, so difficult to counteract, and so difficult to treat. So here we have a visual of what citrus greening looks like with our plants. On the right-hand side, this is a basic visual of what a leaf would look like as a result of getting infected with CLAS. A blotchy model is what they refer to it as, where we have unregular patterns forming on the leaf, these yellow blotches on the leaf that are pretty much asymmetrical, is what you would normally see on a new leaf that's formed in the citrus. So when a citrus plant gets infected with CLAS, usually the newer leaves that are growing throughout the lifespan of the plant is what you'll start to see the effects of CLAS going into. The blockage of flowing tissue due to movement of bacteria causes a hindrance in the nutrient flow. So that's why we get this discoloration. Eventually, as the disease progresses, we do have leaf loss, and ultimately the plant ends up passing away. Other visuals that we can see due to citrus greening is uneven fruit size. The taste of the fruits ends up becoming very bitter, and lopsidedness due to I imagine it's also due to hindrance in nutrient flow. And so we'll see a little bit more visuals on the next slide. So this is a visual provided by Tiku and his research department. that I got off of the database. On the left-hand side, we can see a normal healthy fruit. And on the right-hand side, we can see how the fruit has shrunken due to citrus greening. And then image B shows how the fruit has a lopsided growth. And you can't really visualize it with this image, but basically one side is going to grow normal and the other side is going to pretty much be asymmetrical. Visual C is another representation of that. And here we can see noticeable notches on the leaf on the left-hand side. So either left or right side of the leaf, it doesn't really discriminate against, the pathogen doesn't discriminate against which side of the leaf is going to end up being notched, but you can see how there's quite noticeable damage. And then this is due to early exposure to sea lice. HLB is also another form of calling sea lice. Well, HLB is the actual disease that was named way back when in China 100 years ago. It stands for Huanglongbing, which is also a translation to yellow shoot disease. A bit of relevant facts, I guess. And on the right-hand side, we can see how the notches, as a result of long exposure to sea lice infection, the leaves that form, the new leaves that are growing, end up becoming very disnatured. They're pretty much inefficient. They don't really produce any benefits to the plant. And ultimately, the leaves are going to fall off and they're going to die. So next, we're going to start talking about rhizobium-mediated transformation. This is one of the techniques that is implemented at the AgriLife Extension, at the research department that we're in. So we basically want to induce an environment that is habitable for sea lice, because sea lice is a very difficult pathogen to work with. It's unculturable in the lab, and it does not survive in very many conditions other than through the phloem. So basically the vascular system of the plant. Here I took an image of what would be presumably a plant with hairy root induction. And on the bottom image, we can see how hairy roots are formed. So basically, we inoculate the plant at the part where we want the root to grow, because of how the plant cell plasticity happens, since plant cells are so malleable and the word is plasticity. Since plants are so, the cells are so efficient at being changed from the stem cell to specific cells that work for specific things, you can inoculate the plant at, let's say, the bottom portion of a branch, for instance, and the inoculation of a bacteria known as Rhizobium rhizogenes ends up introducing its transfer DNA, root-inducing transfer DNA specifically, and that causes the plant cells that are in that area to take up the gene and modify itself to start producing hairy roots. So basically what hairy roots are roots that normally, when you have a plant, they don't have the same vasculature that hairy roots would have. Basically, hairy roots, they gain phloem and xylem capabilities. They have intact phloem and xylem, so they gain that vasculature that sea lice can inhabit, making it the most, let's say, optimal form of maintaining the sea lice. As roots in general, you can chop them up pretty much and they'll grow back. Same with leaves. So other aspects of hairy roots that I didn't add into the slide is that they like to grow upwards instead of downwards. As you can see, you can kind of see in this image in the bottom, they have this specific detail, I guess, encoded where they are anti-gravitational, so they do like to go against gravity and grow upwards. And the genes that are encoded into the actual rhizobium, the actual microbe, is called the root locus genes, and these are the ones that form little tumors on the plant. We basically modified the bacteria to not be invasive towards the plant, but also produce that beneficial hairy root that we want. So they do form little calluses at the bottom of the branch. And in this case, this is where we inoculated the branch so that it could start forming roots. And this image doesn't really show it very well, but there is a little callus that forms, and that's where the roots originate pretty much, the advantageous hairy roots that we want. So here we have an image on our little methods of inoculation. We basically culture the rhizobium microbe, and then we smear the branch or the leaf that I also implemented in the bottom. Right at the petiole of the leaf, you can also inoculate it. So on the top, we have branches, and we put them in vermiculite soil, and we constantly mist them every 5 minutes or so that the environment for the rhizobium rhizogenes is favorable, and they can produce their roots roughly between 2 weeks to 4 weeks. You'll start seeing root formation in like around 2 weeks in. So down here, this is just the way we inoculate the leaves and then plant them into the vermiculite soil. We chop up the top part, as you can see here, because another technique is used that one of our coworkers is doing, but that's for another time, I guess. But basically, this is how you can visualize how we inoculate the hairy root and plant it and place it into the greenhouse. It's very interesting to see how you can actually use leaves and grow roots from the leaves using the petioles. This is going back to how cells, how plant cells have such high plasticity. You can basically genetically modify them to do anything that a plant would normally do. You could pick any stem cell, genetically modify it, and whatever you want that stem cell to do, the plant can produce it. So here in this slide, we have hairy root identification. We use a green fluorescent protein, GFP, as our marker. And on the bottom down here, I have a typical transgene cassette, CRISPR cassette that would show us how we splice the gene and enter it into the rhizobium genetic makeup, and then that rhizobium genetic makeup, what it does is the plasmids that it produces can get absorbed by the plant. Basically, that's why we pick rhizogenes in particular, is because they have the potential. Well, they have the potential of being able to be taken up by the plant, because not every microbe can transfer its genes over the same way that rhizobium can. So here we have on the yellow portion our promoter gene. That it's labeled as gene just as like as a general term, but that's basically the gene that we want to implement into the plant. Is where we would splice it. We have our 5' cap and then our 3' cap. And then our GFP would just be our marker, just to be able to tell that the genes that we want, the hairy root genes that we want, are present in the actual root. So on the top image up here, we have what a non-transgenic root would be. So this basically means that the roots do not have intact xylems and phloems, and they would not be able to maintain the CLAS pathogen. And then on the bottom, we have an image of what green fluorescent protein would look like under a fluorescent microscope. And these are basically what we would call hairy roots. These are the ones that actually have the vasculature necessary for maintaining CLAS. And these would basically, I didn't really get to this part of, I didn't have time to, throughout the span of the research program, I didn't have time to really fully in depth go into this and practice it myself. But the idea is that once we gather our DNA from the transgenic material, we would then implement it into a PCR analysis. So either PCR or qPCR, which is our quantitative polymerase chain reaction analysis. That's mostly what we focused on my portion of the lab. And the diagnostic tools would basically be looking for either the genes, the root locus genes, or the actual CLAS present in the plant, depending on what we're trying to detect at the moment, because it is very difficult to induce hairy root into the plants. The actual percentage of plants that can, the actual percentage of transgenic material that we produce is kind of low compared to the actual CLAS infection. And so it can be pretty difficult. It depends on a lot of different factors, and timing is also one of our biggest difficulties in the lab, since we work with plants that take very long to grow and produce what we are looking for to experiment on. It could take up to 6 months, 12 months, all depending. And then with our low yields and our root locus genes that we're looking for, it could be very difficult. But basically, on our right-hand side, we have a graph of what a normal qPCR would look like. This would be the data analysis. I use an example from a previous research publication from our very own lab. So on the left-hand side, our Y value would be our normal CT values or CQ values, which would be our cycle threshold, our cycle quantification measures. These values would basically tell us how much of the either the CLAS pathogen or our rhizobium genes are present in the plant. Usually we want a range of between 20 to 30. In between those two ranges is how we would be able to detect. So basically what CT values are cycle thresholds in which the necessary amount of reactions needed, or the necessary amount of cycles in multiple reactions in PCR actually are needed to produce a fluorescent signal. Our GFP marker would be that fluorescent signal in this case. And a low CT value would mean that we have high fluorescent signaling, would also mean that we also have high hairy root formation. And a higher CT value would be the inverse of that. So basically, it would take more reaction cycles to be able to send that signal to us and tell us that there's hairy root presence in the plant. Our control genes that we use for the plant is the GAPC2 endogenous genes. These are basically just controls that we measure to compare and contrast between our values and our transgenic material and non-transgenic material. And other than that, we use specific markers for ribosomal proteins and and we also use the ribonucleotide reductase beta subunit genes. This is for CLAS estimation. So we have our root-inducing genes and our CLAS. We can use qPCR to quantify measures for both of those. Conclusions that I implemented on this slide is I just wanted to talk about an overview of hairy root induction, its prevalence in the plant pathology field, and also talk about how CLAS is still an ongoing battle that we are trying to ultimately overcome. So our rhizogene-mediated hairy root induction is beneficial in gathering data. Basically, since we know that rhizogenes are a more preferable place for CLAS to form, that's non-invasive to the plant. We don't have to cut off branches. We don't have to hurt the plant necessarily. Roots are relatively easy to work with, and they grow so fast. The plant, you don't really have to worry about any damage to the plant as a result of chopping up a whole bunch of roots, for instance. And one of the most important things in research in general is data. So qPCR measures, DNA extraction, other PCR measures, and diagnostic tools are so beneficial to research as a whole in gathering what works, antimicrobial genes, for instance, or let's say maybe other genes that we can implement into the plant to build a resistance to not only CLAS, but any other pathogen in general. And I think that's all I have for this slide. My acknowledgements, I wanted to thank the Department of Homeland Security and CBTS for providing this privilege to be able to experience with y'all. I also wanted to thank the AgriLab Research Extension and West Uphill for also giving me this opportunity. I wanted to thank Dr. Mandati himself here and a couple other coworkers and mentors. So Rajita Kaburi is one of the postdocs, and Aditya Kolstrata. He's the one that helped me out the most with my work, and also Victoria Mora, who was my mentor throughout the whole research, the whole project that we did over there. And these are my references, but other than that, that is my presentation. So any questions? Sorry, any questions?
Good. Maybe I can ask a broader question. So how do you think the tools that you learn in the lab could help with a career, and like you want to go non-plant related?
Oh, that would definitely, well, just in research in general, my major in particular is biomedical science, and there's, at least in my instance, I haven't been able to find very many applications other than research for the degree that I got. So maybe humanitarian research, public health. Since we can use PCR, qPCR, real-time PCRs in any aspect of science relating to proteins, genomic makeups, pathogens, and all of that, that would be that is very beneficial now, even though we probably developed these tools 40, 50 years ago, we're still implementing them to this day. And that's something very interesting to look at, how we've only expanded and improved upon these tools and added more to what we already have in correlation to research as a whole. So,
Yeah, next. Thank you, Max.
thanks everybody for listening. Perfect.
All right, good morning. Thank you for coming and stopping by. And my name is Axel Rodriguez. I'm an undergraduate in biochemistry. And I had the pleasure of working here in the biochemistry, or in the Korolsky lab here, where I was able to develop skills working on different experiments and learning different methods or protocols or techniques and the instruments we use, which allows us to both directly and indirectly detect pathogens. So in this presentation, I will express the experience I learned and pretty much connect how we can, excuse me, in this presentation, I will express what I learned throughout this experience and how we can apply it to detect and neutralize the pathogens, the biothreats.
Water.
No, I'm fine. Thank you. Okay. So here I present the idea, the illustration that we want to create monoclonal antibodies to detect these biothreats. However, these antibodies on themselves, it would make it difficult for us to detect these antibodies and make sure they reach their target pathogen. So we have the idea to create to attach these antibodies onto the nanoparticles. That way we can enhance the sensory output of these antibodies. So as a preliminary experiment to this idea, I did a study on transferrin TTR sorry. So preliminary to the experiment of creating antibodies, I first took the experiment of studying TTR and studying its structure and Thank you. Yeah, I'll take some water. Read it.
How about your friends? They're all friends.
If I drink too much coffee, maybe that's it.
It's
honestly.
If you wanna walk, stand up and do it, it's easier. If you're uncomfortable, see?
Yeah, I was like, all right. Sorry about this.
I'm good. Everybody goes to the stage.
Yeah. All right. So the idea is to create monoclonal antibodies to. So these antibodies, we want to attack and target the pathogens and eventually neutralize these pathogens. So as a preliminary study, we took TTR to make sure that the nanoparticles wouldn't disrupt the structure and stability of the protein. So as just a brief introduction to TTR, it is a tetrameric protein that is synthesized in both the liver and choroid plexus and a few other regions of the body that will transport thyroxine, a thyroid hormone, and retinol binding protein, which will later bind and transport retinol throughout the bloodstream. So here in the image, or in the illustration below, we have an example of a protein, a tetrameric protein that can, under certain conditions, can dissociate into monomers. And these monomers can undergo misfolding, and this misfolding can eventually result in the creation and clustering of what we call oligomers or aggregates or fibrils. So moving on to the primary instrument that I utilized for this experiment is the atomic force microscopy paired with infrared spectroscopy, which we'll just call AFMIR or nano-IR. And this is an example of direct detection for pathogens and other molecules. So, of course, so the specialty of AFMIR is it can both image the sample, and we can pretty much, like, it's a single particle characterization of this molecule or whatever you are studying. So I'll show it in just a second, but the theory here in the second-hand image on the bottom pretty much shows us that the tip of the probe can pretty much, we can get like a sample all over the place. So we can pinpoint an exact location on the sample, and by doing so, we can pretty much the theory is shoot laser and create a thermal expansion on the, in this case, a protein. And we can acquire a spectral analysis doing so. And it does it by, if I'm not correct, or if I'm not mistaken, it'll, by the thermal expansion, it will acquire the frequency of that thermal expansion. And in this case, we were studying the secondary structure of the protein, since that is the most, I guess, one of the, it is the indicator for the aggregation stage for that fiber, or sorry, for that protein. So, in this, for this experiment, we took the protein and we grew the protein first in a broth. And then once we collected that protein, we collected the protein and we put it in a buffer solution of a pH of around 3. That way we can drive the aggregation process. And we took different time points ranging from 0 hours to 48 hours, and we took 7 samples in total. And after that, we collected the sample and we will deposit it onto a gold nanoparticle substrate, which to me is honestly the hardest part of anything I've done, because especially when I drink caffeine, like I have shaky hands. So it's a good thing I'm not going to become a surgeon. And so yeah, keep in mind it's tiny little like substrate, and I've done pipetting work. It's not too difficult, but keep in mind that like I had to hold it with tweezers and then hold it with my pipette, like not great. But I got better along the way, and it's great. So yeah, so that's it was a great learning curve in practicing how to pipette very precise microliter quantities onto a tiny little substrate. And then keep in mind that I have to pick it up, and then with a second set of tweezers, you have to clamp it so you don't contaminate the sample. You have to rinse it out with DI water, and then dry it out, and then place it on a metal disc, and then ready to go. But before it's ready to go, I had to learn how to optimize, or use the instrument pretty much, and I had to optimize both the probe and the laser before we could even analyze the sample. And that can range around 20 to 40 minutes, just optimizing the instrument itself. And then we can then analyze the sample, which brings me to the next point. So here, on the right side, I'll begin, I should say them, but on the right side first, we have the examples of imaging used through the AFM. And on the left half of those images, we have a 3x3 in micrometers, and just a few examples of different time points. Just the first top is, if I'm not mistaken, 3 or 6 hours of this protein aggregation. And we can pretty much take point spectra of each particle that we want to identify. And then on the right side of that figure, or of that image, we can take an even accurate, a more accurate, like precise, or we can take a more precise image to get more accurate results. In this case, it was a 0.7 by 0.7 micrometer. On the bottom, same concept, just different time point. And then, water. And after imaging, then we can take a point spectra of the aggregate again. And then on the left-hand side, we have a figure that which will quantify this vibration caused by thermal expansion. And then we'll average out the spectra that we acquired throughout the time points. And here at the 1630 to 1674 region, we consider the amide 1 region for proteins, which is, we can relate to the secondary structure. At 1630, we can identify that it is the parallel beta sheets, and at the middle one, 1660, at around 1660, is the alpha helical and the random coiling of the secondary structure. Lastly, we have the 1674, which is related to the anti-parallel beta sheets. So here's just a bit more, just quantifying that spectral analysis by analyzing the area created from those peaks. And here we just see like an example of 0 hours to 48 hours and see how those secondary structures differentiate between those time points. And here we can see, in this case, I guess, real briefly, the alpha helical structure and random coiling is dominant compared to parallel beta sheets, since they're just oligomers. And this allows for direct analysis. Now moving on to the second experiment is using Raman spectroscopy, which the other one was AFMIR. This allows us for indirect detection for pathogens and other types of conditions. In this experiment, our initial plan was we grew, as you can see in this picture, we grew little baby sprouts, and we grew these rice in soil in little pots instead of hydroponically, since it would be quicker and also be easier to apply the stresses, I should say. And so the stresses, we had abiotic and biotic. So the abiotic stress, we added different levels of arsenic to these plants to induce the stress. And then once that these plants would grow larger and produce leaves, we wanted to inoculate fungi onto these leaves to induce the biotic stress. And then using the Raman spectroscopy, we would use a laser or use the Raman spectroscopy to identify the changes in its structure and so unfortunately these sprouts did not make it all the way through. The stress was a bit too much for them to handle, and we did not make it to the end to produce results and gather the data. But in previous cases, or in the Kudowski lab, we have previously worked with corn and wheat and different other types of plants, where we have induced these abiotic and biotic stresses, so similar conditions, and have proven that Raman spectroscopy is able to indirectly detect these pathogens through different components in the plant's composition, which we characterize with certain compounds such as carotenoids, pectin, and cellulose, which are usually for the structural components of the plant. So as a conclusion, again, with nano-IR or AFMIR, we're able to directly detect the pathogen. So in this case, we just analyze the secondary structure of the protein to secure that these nanoparticles do not affect the structure, the structure of the protein. So in this case, we can now apply the antibodies or in any relation to this to then target and eventually figure out a way to neutralize these biothreats. And then the second part is we can indirectly target and detect these pathogens and biothreats through Raman spectroscopy. And overall, it was just a great experience in learning how to use these two instruments, I should say, and the whole protocol, growth, analysis, everything, and just great overall. So real quick, I just want to thank everyone, especially the Kudowski lab, for being by my side, helping me out, the analysis, and being there, getting food, as always, and of course, the DHS fellows and personnel at CBTS. Great experience. Sorry, I'm not like a huge speaker, so always in my mind, I'm like, I'll get this done, and then if I have something planned, it's like never works out. But if you have any questions, feel free. I'll do my best to answer those one-on-one. Send me an email, ask for any contact information. I promise I'm not as like. Sorry, anyways, too much caffeine. I'm sorry. Any questions? Sorry.
Yes. And the IR graph where you characterize like the percentage of parallel and anti-parallel sheets, are those just all oligomers across?
Those are just oligomers. There were no firewall characterization there.
So you said the rice plants died, like as an.
Unfortunately, they did. They did not make
To
it.
kill the plants.
It was too much fun.
What was it? Yeah, arsenic killed the plants, or was it was fungus?
Since rice is better grown hydroponically, I would say that. I mean, the soil isn't bad, but I think since it will just like stay like the residue will stay, like the arsenic residue will stay in the soil. It was just too much stress for the plant to handle all at once. It just did not make it past.
So the other way to do it maybe is to grow them up and then add it after a while, maybe
could.
they could tolerate better.
We could grow them up to like a certain time point, like say like once they start sprouting and maybe some leaves, we could then start applying the stress to see if the leaves turn brown or like if they start dying off, and we can apply, we can inoculate them at the same time and see how that goes. So yeah, it's a good idea.
You got some experience in handling plants.
Growing plants. Growing plants.
We're not farmers, right? But you know, we're not farmers, you know, that's why you can appreciate how farming is an important thing.
Oh, yeah.
You know? It's not easy to grow plants in the greenhouse.
There will be just pop the seed in there and then it'll sprout, and in this case, like, it will, but you have to like manually just keep watch on it. It's like a little baby too. Any other questions?
It's a big question for you and maybe also Dimitri, but in the graph it showed the like increase in the helix and the like unsigned coils. Why is
About the 9-hour one, the blue
yeah,
one?
because and then it just goes back to the same level at hour like 12, as it was at like hour 4.
Alright, so the analysis here, or the interpretation we can take from here, is pretty much, so these monomers will aggregate, or they can cluster and form oligomers, or form aggregates that are either characterized as oligomers or fibrils. And in this case, oligomers are just like, I guess we can detect them as like spherical structures. And usually these monomers, they will contain a higher intensity of alpha helical structure and mainly random coiling. But in literature, it states that usually lipids tend to drive like the formation of fibrils or oligomers. So in my theory would be that at 9 hours, that's when it starts forming fibrils and also oligomers. But since it's missing, it's lacking like certain like additives, like certain lipids or certain like whatever else, it can't drive that process to create more of the oligomers or fibrils. So then it'll just kind of like dissociate again back to monomers. Like I was saying, like it's not that it can't form it. I'm saying it just like helps it form, so it'll push it forward to drive it. If that makes sense. And maybe if you want to add on to that, Dimitri.
I think you, yeah.
Or TND, yeah.
Okay, so I think it's also very interesting. Usually for protein aggregation, the process is monomeric protein will get into oligomers and oligomer forming fibrils. And for 9 hours, we think it's very interesting because that's actually the takeout point where we start seeing a lot of fibrils. So this analysis actually is focusing more on oligomers, which at least my hypothesis is at 9 hours, since we observe some fibrils, but still mostly oligomers, though those oligomers are at a stage in their late stage elongation where are waiting to be forming fibrils, whereas after 9 hours, for all those aggregate we observed, like 12, 24, 48, which we thought that the fibrils should be most dominate, those are the aggregates that remain as the form of oligomers. And that's why we see random coil and alpha helical more dominant, because they are not tending to form fibrils anymore. And we are trying to do more analysis on fibrils to sort of understand what exactly is going on between the oligomer and the fibrils after they already get through the elongation stage. But that'll be my explanation here.
But are you senior and graduating, or
Yes.
what's coming up?
Oh, I'm graduating. Yeah, this is actually next week. But I applied to a few master programs, so I'm just waiting on those responses.
Environment or where?
In a few different like regions, so like nanomedicine, pharmaceuticals, and also like biomedical stuff, biological sciences. That's my initial plan right now. And keep working, Dr. Korolski, keep it up. And then after that, I hope to apply to get a PhD program here in biochemistry and see where that goes.
Right. Thank you.
Victoria.
Oh, yeah. But I didn't want to.
Also, the exactly like that. So maybe that's. Also decaf, it was decaf, I think it was decaf. Like mine is like, it was too speedy. Like very.
Yeah, right there in the green. Back here? Yeah. Yeah.
Oh, yeah.
Actually, yeah.
That would be more.
It's like easy access to get like a whole background information. And it's like, and like, so like, you can like, explain like which part cross the data.
Hey, they're most charged. They're trying to charge.
It's more like if I.
So what's the next step?
Yeah.
You get another dog. You get the body policies to put it in
Calculator response, I think of that.
I feel that's the way it's working. It's the errors.
That's why I tell students, memorize your.
I'm able to open it. I can do. Hey, everybody, sorry, we gotta keep going. We have another summer presentation after these.
Yeah.
Yeah, go ahead. Introduce yourself and take it away.
I'm Jocelyn. I did a little project on a plant called Nicotiana bentamiana. This is my assignment. So we infected with the plants with the virus called the tobacco mosaic virus, which is also called TB. It is a positive single-stranded RNA virus. It's very highly infectious, so plants can get contaminated with the virus just by a little touch. So symptoms, include discoloration, wilting, and necrosis, which is where the plant dies. And then the actual plant is very popular in the research industry and agriculture. It's basically like a little model plant, I guess you could say, because it's been so successful with other viruses. So since the plant and the virus are very easy to work with, so when you combine them together, it's like magic, basically. So what I did was to observe the physiological symptoms of the plant at different time points, and then molecular characterization by doing RT-PCR. So here we just had my seeds, the soil that we use, and then we plant them, water them, and then this is the germination after 1 week, and then at 4 weeks is where I started the infection. So then from there we rub inoculate the leaves. So we use previously infected tissue from another project, and then we used 250 microliters of the buffer with the previously infected tissue, and then we just rub inoculate it, just rub it on the leaf. And then from there, we have to prep the samples. We ground them with liquid nitrogen. So you have to really get in there and make it into a fine powder. The very end, it's protein powder. Yeah. And so then the RNA extraction kit that we use is called Direct Sol RNA Mini Prep Plus. So then we start with the lysine of the cell. So we use Trizol. And then we just go on. And then we do RNA quantification, which is putting them on the nanograph. And then we just run it on a gel. And then we have the gel, which is basically tells us how the quality of the RNA is. And they're okay, not the 48-hour one, because we use 0 hour and mock, which is healthy plants, that makes sense. And then we observe them we observe the plants at 24, 48, 72 hours, and then 7 days post inoculation. So they're all pretty okay except the 48 hour. Not sure what happened there. The RNA could have been degraded, but. And then from there, since with like gel electrophoresis, I think that's how we pronounce it. We can't use RNA, so we have to convert the RNA to cDNA, which is complementary DNA. So we use the random primer, which it like targets random parts of the RNA or cDNA converted to cDNA, which is called reverse transcription because DNA turns into RNA, but then we're turning it back to cDNA, so it's reversed. And then from there, we run it on a PCR, which basically is just amplifying the cDNA. So you have your template right here, and then in denaturation, the strands like separate, and then in annealing, it cools down by a lot so that the primer can like attach itself to the cDNA template. And then in expansion, it heats up a little bit so that the tag polymerase can like extend the primer, which by adding like new complementary nucleotides, and it just goes on and on and on. And then the cycle repeats for like 30-ish, 35 cycles, which lasts like an hour and a half, maybe. And then for my results, okay, so here we have like the 24-hour mark. There's not really much of a difference, but then at the 48 hours, you can kind of see like little blotches of like mosaic, which is like the discoloration. You can kind of see it. And then at 72 hours, you can see it a little more, but it's okay. And then at 7 DPI, that's when the plant started to wilt, which is ultimately what we wanted, I guess, yeah. So there was no necrosis. If I would have gone to observe the plant at 10 days, then maybe there would have been necrosis. But at this point, the plant was just wilting, so sad. And then with molecular analysis, when we ran the gel, we used 2 different primers, a replicase primer and 18S RNA primer, which is like a housekeeping gene. So here, it's okay that there's like no bands. That means the hypothesis was right. And then at the 24-hour mark, well, if you can like kind of tell, I don't know if you could, but as between the 24-hour and 72-hour marks, where's the mouse? The bands get brighter and brighter. That means that that there was more of the virus in the plant. And then at 7 DPI, the bands are a little brighter. a little less brighter because the plant is like dying already. So there's like less of the virus in the plant since it's like dying. And then here, this bottom one, it's the 18S primer. It's basically like an internal control. So all the bands are like supposed to come out. It just serves like as a internal control. And so the conclusion is that basically what I talked about in my introduction kind of like ties into it. So since they're both very easy to work with, and the Cochiana bentamiana is very susceptible to viruses, it can be used. Well, it's been used with research because since since it can transfer the virus by cell to cell, it can cause damage to like a lot of agriculture, like crops and stuff. It can be like in the soil, which is very, very bad. So there's a lot of research with it to like prevent. Ultimately, what we use is to like not cross-contaminate, and then we go and we like autoclave the plant so that virus can just gone. Yeah. I'd like to thank everybody, especially Dr. Granti and everybody that helped me, mentors, Rachita, Victoria, and Tia, and of course you, Chris, and everybody that helped me. Does anybody have any questions?
I wanted to ask, so you mentioned that the RNA is turned into cDNA by random primers, and then the cDNA is like, goes through PCR. Are there like specific primers that target the cDNA?
We use a random hexamer, which is
For the PCR stuff?
For the PCR stuff.
for the
after you get
PCR, we
cDNA.
use
Is
for the PCR we use the it's called turbo, the primer that we use, which is like this gel that we ran. Yeah, so we ran that one. It's called Turbo. It was made by my mentor Rachita. And this bottom one is the bottom one is the 18S primer, which just serves as like an internal control. So it basically like kind of proves like, okay, yes, this is Nicotiana ventamiana because the housekeeping gene basically means that like it's supposed to be in the plant. Like we have our own, but the plants also have their own. So with the gel, and it's also present in the 0-hour and mock because it's in the plant. If that makes sense.
No, I get it, yeah.
Yeah.
Thank you.
So that's the replicase is the specific primer that for the virus itself.
Yes.
Yeah.
Okay.
Yeah, so at 70 PI, the bands are like less bright because the plant is like essentially already like dying off. So that's why.
You still see very, very good fibers in there still.
Yeah. It's bright enough, but it's less brighter than at like 7.
Peak is 72 hours.
Yeah, the peak is 72, but that one is like the brightest. Because there's more and more of the virus present in the plant. And then at 7 days post, the plant was already like dying. It's already like routine and fast. I can't remember.
So you mentioned, just to switch on the same topic, but the virus infects, this is a model plant,
Yes.
Ventamiana, tobacco. What are the agriculturally relevant crops that this virus affects?
Oh, and it can also affect tomato and spinach. There's a lot more. I can't think of them at the top of my head. I just remembered tomatoes and spinach. And tomatoes, it can cause like yellowing, like around like the tomato and stuff. And if it like touches another tomato, obviously it's gonna infect that one and just goes on and on and on, which is like very bad. Because that like affects the crop yields and that's when we can eat. The tomatoes are infected, then we can't eat them. And, well, you can't really kill the virus unless you like,
Basically, remove the virus.
yeah.
Throw it in an autoclave.
Throw it in the autoclave and you're good. But you can't do that with everything. Yeah. This is just the way.
Something like the
I'm going to ask you this question. What is your future career plan?
Oh, me? Oh, so I graduate in December and I'm gonna take the year off to work, save money, and I'm gonna apply to a master's program in anesthesia. Super great. I hope I get in. So that way it's the career is called an anesthesiologist assistant. It's kind of like a PA, the anesthesia department. It's only a 2-year program.
Justin has figured out the next 10 years in life.
Yeah, seriously.
She's one unique person that she knows where to go, like 3 years, 4 years, it's school, and like she's in that path.
Yeah.
This is a detour for us.
No, I'm not working out a lot.
Detour and squat from that.
Yeah, I love working out a lot. It's so easy.
No, I can feel that, yeah.
But yeah.
Apparently, there's only 2 anesthesiology schools in the entire Texas, so.
Oh, yeah. Yeah, it's in Houston and Austin, so I have to apply out of state. But
I mean, we don't have a random here.
no, I checked and there's none. So in Houston and Austin, so I have to apply out of state. Some of the out of state programs are cheaper than the one in Houston, so even with like out of state tuition. So yeah, that's what I have planned for like the next 5 years. Yeah. Thank you.
So the best for the last.
Yeah.
Happy days.
The happy sisters. I hope they're happy.
Sorry.
Hey.
That's not a happy cow.
He sounds like he's smiling.
That's a cute cow.
It's a cow, but it's scared.
Oh, nice.
All right.
That would be good.
And don't forget to introduce yourself too.
Let's see this. Okay. Yeah.
Funny, yeah. I have to get out of behind you.
Yeah, it's always the way everybody should get out of there. Anyway, so my name is Aiden Holman. Today I'm presenting on the identification of adult exhibited ticks by Robin spectroscopy with species. Let's get into it. Just a brief background. So tick surveillance is crucial for controlling tick-borne diseases in cattle production. So right now, cattle that come into the U.S. Cross the border through a very long and terrible process. I say terrible because it's long. But they have to, like, as you can see, a lot of cattle come through there, and then they have, like, one by one, they have to be put into that little contraption? Yeah, that's the word. And then searched up and down in every crevice for ticks, for signs of ticks. And what's difficult about that is that tick sizes can actually be too small to feel sometimes. If they're not feeding on the cow, that means they're usually going to be too small to feel, and so we can't find them. And that might be an issue because heart ticks, the one that we're talking about, they leave the body 3 times in their life cycle. So if they're not there, but they were there, the pathogen can still be inside that cow. So we need another way to identify to see if ticks were there and which ticks were there. Another important thing about this is that tick-borne diseases, such as cattle fever, pose a significant threat to cattle health and production. A single cattle fever tick outbreak is predicted to cost over $1.2 billion within a year of surfacing. Luckily, Raman spectroscopy shows promise for identifying tick species through their feces. Raman spectroscopy is itself the measurement of inelastically scattered photons from specimens under visible light. Our methods, so we've scanned 57 samples of ticks from multiple genera. Amblyoma, which is the tick genus that vectors pathogens that cause things like Rocky Mountain spotted fever, rickettsial pox, and other rickettsial diseases. We have Rhipicephalus, which is our cattle fever tick that I talked about earlier. Dermacentor, which also vectors pathogens that causes Rocky Mountain spotted fever. Haemaphysalis vectors pathogens that cause tick-borne encephalitis. And Ixodes, which hopefully some of you are familiar with, it vectors the pathogen that causes Lyme disease. Within those those 5 genera, we have 12 species total of ticks, as well as 2 additional samples of hornflies. Hornflies are important for this because hornflies also, they show up just as often as ticks do, if not more often. So if we're looking at feces on cattle, we want to make sure that we're identifying tick feces and not other insects or arthropods. So we use a machine learning model called PLCA, which is partially square discriminant analysis, which is discriminant analysis. We use it to test the feasibility of a model to accurately identify individual spectra of the correct class. I have an example for y'all. Let me see, they're actually hard to point to. So if you're
The pointer?
You just go below.
Oh, that's right. Yeah.
no. Oh, yes. They're just very slow. So coming from the top, you have the number of spectra that are actually this class. So this will be actually oranges. This will be actually apples. coming from left to right, you have the number of spectra that were predicted as that class. So right here in this little box right here, we have 20 spectra that were actually oranges, were predicted as oranges. And so we have a true positive rate down here. Mouse go. The mouse disappeared there. So that'd be 20 out of 25, which is 80%. We had 5 of them that were predicted as apples that were actually oranges. And so we have a false negative rate of 20%. On the right side, okay. On the right side, we have 2 of our actual apples that are predicted as oranges, and so we have an 8% false negative rate. And then we have 23 out of 25 of them that were predicted as apples, as being apples, and that was a true TPR of 92%. So that's a pretty good model. It's a big model, but it's a pretty good model. Tick frass collected from the skin or fur cattle and sheep from institutions in Oklahoma, Washington, and several in Texas. We utilize a confocal Raman microscope with a 750 nanometer laser. Joseph, who was the first presentation, had like a picture of it with a pretty blue laser. We have a red laser, so it's not as pretty. You can remember that. We acquired 20 to 25 spectra from each fecal sample with 10 to 25 second acquisitions. Looking into our results, so beautiful colors are going to be our tick genera, and our top gray ugly little spectra is our hornfly, which is Hematobia irritans. So our bands, which are basically the Raman shift, so bands correspond to like the 1 Raman shift, so 600 is band. Our black bands are going to be the guanine, so on something guanine, that light hit reflect, and that's going to be specific to guanine. The red bands are going to be blood, so hopefully that's easy to remember. It's not blood, it's components that belong in blood that we're seeing. But yeah, as you can see, some of these don't have don't have peaks at those bands, and so we call them blood-like species. And then some of them that have like just basically just guanine, or we'll call them guanine-like species. And we'll get into why that's important too. Oh, and I also wanted to point out 2 of our specific blood-like bands belong to human groups. So our first PLSDA model is going to be our ability for PLSDA to predict genus out of the multiple genera that we had. So amblyoma, again, is the vector that carry pathogens that cause things like Rocky Mountain spotted fever and rickettsial diseases. We had 100% accuracy of our model to predict that using Raman. Rhipicephalus, remember our cattle fever, the one that cost us $1.2 billion in a year. We had an over 98% accuracy to predict that one. Dermacentaur, we had over 93%. Haemaphysalis, we had over 100%, and then, or 100%. And then Ixodes, we had 95%. We also want to see if we could predict the species from each genus. And so we had 3 species in the Rhipicephalus and the Dermacentaur. In the Rhipicephalus, annulatus had 100% accuracy. We had 95%, over 95% for Microplus, and then 100% for Sanguineus. What's cool about this? Well, what's not cool about this one is that all of these will carry or will vector the pathogens that cause cattle fever. When we get to the bottom one, Dermacentaur. So Andersoni is actually the only one that we see on cattle, basically. Albopictus is not so much. Variabilis is going to be our dog, our American dog tick. So we see that on dogs more often, but there are reports that they have been on cattle. So the Andersoni and Variabilis are the really ones that we care about. But it'd be cool if we see Albopictus on cattle. That means we have now a report that they are seen on cattle. But they do also carry different pathogens. Andersoni is our main one for cattle fever, Rocky Mountain spot fever. But we have 100% for Albopictus, 99% for Andersoni, 100% for Variabilis. Back to our hornfly versus ticks, you can see a great difference. Our red is going to be all our average spectra for ticks, and then the green is going to be our average spectra for our hornflies. Again, they are very different, but they do share peaks. We had a peak at 653, which is going to be our guanine-like. And then we have some other peaks with the bloods. And for our PLSDA, we had over 98% for ticks, to divide them between ticks and flies, and 100% for flies. So that was pretty good. Coming into our discussion, so Raman enabled accurate identification of multiple genera, like I just said, over 93% accuracy, and species with over 95% accuracy. Raman also enabled accurate differentiation between ticks and hornflies with over 98% accuracy. We noticed that hornfly species were not as similar as we thought they'd be to blood-like tick species, and we have some explanations for this. So we found that a female D. andersoni tick, which is the one that has the blood-like tick species, it can engorge up to 4,000 milligrams of blood in one sitting. But a female hornfly will imbibe, which is like, because they have spongy mouth parts, 1.7 milligrams per feeding event. So that's a big difference for us, and so we're thinking that we're seeing more of those peaks for the blood-like bands because there's more blood being wasted. I think that's the word. Pooped out, yeah, science, yeah. Additionally, which might be a little obvious, is their digestive processes are different. For example, insects' primary waste product is uric acid, while ticks' primary waste product is guanine. Anyone who's confused, ticks are not insects, they're arachnids. Future directions, and so I have some entomological directions because I am a major in entomology, so you can close your ears if you don't want to hear this. But we want to use gas chromatography and mass spectrometry to more accurately evaluate the compositions in our spectra. And we want to make these comparisons to see if ramen can be used to quantify some of these components within our feces. We want to do it all across all 12 tick species, of course. We also want to check for end-species genetic flow variations in ramen spectra. So a tick species from one area of the United States can have different genetics for the same tick species across the United States somewhere else. So we want to see if that also fluctuates our ability to predict those species. We also want to check for variation between male and female fecal spectra. Females do feed on blood, more blood than males do because they have to have that nutrition to reproduce. Yeah, and so the ones that you care about, and that's 3 directions, we want to see if our spectra are comparable to the spectra that we got from our control are comparable to like a handheld Raman spectrometer for easier field-based applications. So you can just take that spectrometer to the field and scan the pieces there. We want to see if that's comparable. If my lecture cited, thank you for the opportunity. More acknowledgements. So special thank you to Drs. Pete Seal, Dimitri Krosky, and Samantha Hayes. They are my collaborators for this project. They give me invaluable feedback, directions. They have a huge wealth of knowledge. Of course, they supply the tick brass, which we needed. Thank you also to Kenny Dow, because he helped me so much with this project, and he's the one who basically got me on this project. And I'd like to thank, of course, CBTS Center of Excellence, everyone there, and also any DHS personnel for the funding to explore this amazing opportunity. That's my space.
So, question.
Sorry. Yes.
What kind of analytes are you hoping to find in GCMS?
So I'm not familiar with GCMS, but I know that the components that we're looking for include things like the heme groups, other amino acids that we find. Can't think of any right now.
Okay,
but yeah.
I was curious.
Question.
Yes.
So ticks carry pathogens.
Correct. And there are salivary glands.
Right. So in this collection that you did you see any already with the the pathogens in them, or
so we're not.
Are these all clean or carrying pathogens?
The ticks.
In fact, they cause tick fever, right? In cattle.
Yes.
Caused by what?
So I'm not collecting the feces. My collaborators are, but they have each of them have farms at different facilities. Like I said, Oklahoma, Washington, different Texas, and they raise these cattle or sheep, and they isolate which ticks are on which. And so when they collect the feces, they're just combing it off from the fur and collecting that. They never reported just seeing any kind of diseases. I'm assuming because they're weird, they're probably not.
Sorry.
These pathogens.
Okay, I wonder if we can differentiate the ones which carry the parasites or pathogens and those that don't,
The pathogens shouldn't alter the way ticks would digest blood. I don't think they do anything like that. They don't really harm ticks as well. So I don't think there'd be any differences because they do stay in those salivary glands. What they're harming would be the animal in the host.
So it goes directly into the bloodstream versus coming out from the feces, the pathogen.
Correct.
Is that right?
Yes.
Okay.
For ticks, yes.
How long do you have to scan? So if you were in the field, handheld, how long?
So handheld is I think it takes a minute for a handheld. But we haven't used it yet.
Okay. Oh, sure. I was just curious.
You're good. I've used handheld.
This is not with the handheld?
No, this is all confocal.
Oh,
this is all used in
confocal. This is the sampling.
Yeah, I
So
want to
you
make
could
sure.
could you use a handheld one?
We haven't tried it. It has a way higher power, so in theory maybe, but it also has a wider laser, wider diameter,
Okay.
which might have complicated things. But yeah, because of how small the pieces is. Yeah.
Yes, last question. How do you enjoy scanning the pieces?
Oh, it smells great. I love putting it in my hands and then rolling it around. I love bathing in it. Yeah, it's really
It's the
fun.
best parts in the lab.
Yeah. I'll do the same.
And maybe in the audience, if anybody has.
Yeah, is there any questions online for Aiden or actually anyone else that
Hey,
presented?
this is Jared. I don't have a question specifically for Adam, but I just wanted to say, because I have to jump off here in a few minutes, that great job and phenomenal work by all the students. As I was probably around the third presentation, I kind of had the sense of relief that, you know, the young adults are doing this amazing work. And things like tick feces, when you tell people you study that, you know, they may think, what's the big deal of that? But all of those presentations, all the work that you are doing have direct impacts in the real world. I mean, in my job, for example, the lady that was talking about the tobacco mosaic virus. Several years ago, tomatoes and peppers, for example, from Mexico were inoperable. They were considered low risk. And so they would be coming through our inspection lots. We wouldn't inspect every shipment because they're low risk. People would bring them through the passenger environments just to use in their homes. And then Mexico was found to have what is called the tomato brown rugose fruit mosaic virus, I think, which is similar to that tobacco mosaic virus. And now those tomatoes and those peppers aren't allowed to come through like they were before. And that has a huge impact on what happens when a commercial importer in the US wants to bring in tomatoes for the grocery store, for example. Well, those tomatoes now, in order to come in, they have to have a lot more inspection. They have to have a lot more certification from Mexico. That all costs money. So that trickles down. In the passenger environment, people that were used to bringing over tomatoes and peppers every week as part of their groceries, they can't do that anymore. And they've been doing it for so long, they forget that it is now prohibited. And sometimes they get penalized because they didn't declare it because they thought, well, this is low risk, I don't have to say anything. So that work is extremely important. The discussion about citrus screening, that project, it's scary that, you know, we're the world's third largest exporter of citrus products. And the introduction of these invasive pests, whether it be the citrusillid or the pathogen, how this detection can really affect, you know, what happens to people's jobs, to people's livelihoods, to markets. It's enormous. I mean, it had me thinking about back in 2003, we had our first case, our first positive detection of what's called mad cow disease, bovine spongiform encephalopathy is the scientific term. But because of that positive detection that we later determined we think came from Canada, we announced to the world that, hey, we have this detection, and the world told us, well, thank you, but we don't want your beef exports until you can show us that this isn't a bigger issue. And in 2004, after all the fallout and the safeguarding and the quarantine, it cost the beef industry $4 billion based on one or two detections of this disease. So that work is extremely important. It is amazing. It's encouraging for me to see the work you're doing, and I just hope that you guys really understand whatever path you go in, that what you're doing there in the labs and what you're going to do have direct impacts in the real world and are very important. So again, great work, phenomenal job, and my best to all of you going forward. Thank you.
Thank you.
Thank you.
Anyone else? Well, thank you, everyone, for a productive summer. We appreciate everybody's time and effort, and know you all worked really hard. So thank you, and thank you for making the long drive from Westlaco. I know the distance up here is shorter than the distance down there.
Right.
So.
No, I really want to speak just for maybe on behalf of also the mentors. Thanks CBTS, DHS for giving this excellent opportunity for all the students. And you guys driving down to Westlaco, that was great. Happy to see you guys more than, this, after this summer, if you wanna come by, give us a holler, okay? So really appreciate again, Jared, also, all the career advice they got, the students, if you're still there, they really loved it. They got to see firsthand, what careers are in CBP and DHS, or even USDA FIS. So thank you all again. And thanks for inviting me to be part of the team.
Yeah, I want to join Grant and thank you guys for setting this program, putting this together. And imagine how much effort it was to, now when we see speakers you invited and how much effort you put to bring us to the Mexican border and show us port of entry. I think it's a great opportunity and it was a pleasure for me to serve as a mentor.
And Dr. Wilkinson unfortunately couldn't come today, but he wanted to say thank you and good work, and he's interested in seeing the slides. So we'll share those as well.
And I want to extend the welcome to visit the lab.
Yes,
if
absolutely.
time permits.
To the tours, yeah.
Yep, we're on the second floor, we can walk you, and then once we're done with the lab, we can have a lunch. We open, maybe some barbecue, please visit.
Sounds good. Barbecue sounds good.
They don't have good barbecue.
Yes. Thank you. Yeah, okay. That's so dry.